Hi, welcome to my virtual presentation titled Quantifying Genetic Diversity of Peanut Cultivars released by UGA using Genotyping by Sequencing. My name is Nino Brown. My co-authors were Bill Branch, Matthew Johnson, and Jason Wallace. So we'll start by discussing genetic gain and genetic diversity and how these two things are intertwined and crucial for crop improvement. And then we'll discuss how we went about measuring genetic diversity of this group of lines and what the results were and what they mean for peanut breeding. So as plant breeders and geneticists, we're all focused and motivated toward improving yield and quality in order to keep up with population growth and product demand. In order to maintain or improve upon the rate of genetic gain, we have to continue to improve breeding efficiency. And that's where the breeder's equation comes in. Delta G, or genetic gain, for a particular trait is a function of heritability, selection intensity, genetic variance, and generation time. A variable that we can modify or leverage in this equation is genetic variance. Otherwise, we can also think of it as genetic diversity of our breeding material. Increasing genetic diversity or variance of populations can lead to higher rates of genetic gain within reason. Crossing wild by elites isn't going to produce new cultivars right away. However, crossing two elites that are genetically divergent can lead to faster rates of genetic gain by improving genetic variance. So in order for us to improve genetic variance within our populations, we need to better understand the genetic relationships of our breeding material. Peanut has undergone several genetic bottlenecking events or reductions in genetic diversity, the most noteworthy of which is the polyploidy event that led to modern cultivated peanut. This would have caused some level of reproductive isolation coupled with peanut's limited seed dispersal. It would have conspired to keep early variation pretty limited. Domestication and early breeding efforts would have also severely bottlenecked diversity. Modern breeding has actually improved diversity of cultivated peanut with the incorporation of PIs with disease and pest resistance, and even some interspecific introgressions such as nematode resistance from Texac 6. So the objective of this study was to determine if UGA cultivars could be uniquely and reliably distinguished from each other. There haven't been many diversity studies lately undertaken with modern genotyping methods focusing on elite cultivars. Peanut cultivars are inbreds, so we wanted to see how diverse they were from each other, but also how variable they were within each inbred line. And we wanted to compare genomics-based diversity measurement to pedigree-based methods. So our entry list included peanut cultivars released by the university since the very early days of peanut breeding at UGA. This included GFA Spanish, which was released in 1938, and Dixie Spanish, released in 1950, as well as several Virginian runner types from the 40s and 50s. And then everything Dr. Branch has released since '86, beginning with Georgia Red, all the way up to the near present with Georgia 18RU, for a total of 32 entries. This group of genotypes represents a range of phenotypes from different market types, growth habits, seed size, seed chemistry, maturity, and more. Recall that one of our objectives was to evaluate variability within these inbred cultivars. So we individually sampled 12 plants from each of the 32 cultivars for a total of 384 samples. The genotyping method chosen was genotyping by sequencing, which was conducted by a service lab, Data2 Bio. Leaf tissue was collected from the 2018 seed increase plots of these 32 cultivars and shipped to the service lab. Once the samples arrived, DNA was extracted, followed by restriction enzyme digestion. Sequencing was conducted on the Illumina HiSeq X platform, and sequences aligned to the recently published Arachis hypogaea genome sequence available on Peanutbase. I won't get too far into the details on the exact methods used to filter and improve upon the data set here, largely because that's Matthew Johnson and Jason Wallace's department. They took care of that part of the project. But I did want to include this slide in case you're interested, want to take a closer look on your own. So the resulting number of SNPs identified in this data set was really exciting. We identified greater than 36,000 SNPs that aligned to the hypogaea genome. 27,000 of those were polymorphic among these UGA developed lines. And looking at this graphic showing SNP density on hypogaea's 20 chromosomes, you can see that the SNPs were pretty evenly distributed across the genome. A couple of hotspots stand out, especially the region I've circled here in red on chromosome 9. This shows the introgression from TexAg6 for root knot nematode resistance. Two of the entries in this trial carry that introgression, namely Georgia 14N and Georgia 17SP. This histogram shows within line genetic variation. It was very limited. Genetic similarity values range from 80 to as high as 100% within cultivars. However, the bulk of individuals within cultivars were greater than 98% similar. Because of this high level of within-line similarity, I combined genotype data to form a consensus genotype for each cultivar. This histogram shows the frequency of genetic similarity values for each of the pairwise comparisons. With 32 cultivar entries, that's a total of 496 comparisons, most of which fall within the 80 to 95% range. You can see that large peak there just right of center, with a small group with greater than 95% similarity at the far right, which we'll discuss a little more later. And then there's two small peaks on the left ranging from 58 to 65% and 70 to 78%, which represent the Spanish and Valencia entries respectively. So the unrooted dendrogram shown here was created using UpGMA. Two small clusters form a primary outgrouping. These are the subspecies fastigiata entries. The Spanish cultivars are circled in brown, and the two Valencia cultivars in yellow. We had really good separation of these fastigiata subspecies examples. We were really happy to see that. We have this big cluster on the right half of the slide, which includes all the hypogaea subspecies. So these are our runners and our Virginias. The first thing that stands out is our modern Virginia cultivars, circled in green here, includes Georgia HiOL, Georgia 05E, Georgia 08V, and Georgia 11J. They form distinct offshoots from the hypogaea group, not clustering with the modern runners. Then circled in red here we have the older cultivar, Southeast Runner 5615, Georgia 119-20, Virginia Runner G26, Virginia Bunch G2, and Virginia Bunch 67. You can see their branches didn't really differentiate very well. They're very genetically quite similar. These cultivars were developed prior to large-scale hybridization-based breeding and were largely developed from plant selection and bulk selections from land races. It is interesting to note here, though, that there's actually a fair amount of phenotypic diversity in this small group, even though their GBS-based estimate of genetic diversity is low. Then we've got the modern runner cultivars, which form three distinct clusters, the first of which is shown in purple there at the bottom of the slide. This includes Georgia 02C, Georgia Runner, Georgia 04S, Georgia 14N, and Georgia 17SP. The second cluster, circled in dark blue, includes Georgia Brown, Georgia Green, Georgia 9B, Georgia 18RU, Georgia 3L, and Georgia Bold. And then the third cluster at the top there, circled in light blue, includes Georgia 6G, Georgia Greener, Georgia 7W, Georgia 16HO, Georgia 12Y, Georgia 13M, Georgia 1R, and Georgia 10T. To compare the GBS-based estimate of genetic similarity to pedigree-based coefficient of parentage, I used the publicly available pedigree information for each of these cultivars and calculated the coefficient of parentage for each pairwise comparison using a program called Winkin. This scatter plot shows the relationship between the GBS-based similarity estimate on the x-axis versus the pedigree-based method on the y-axis. The r-squared value of 22% is significant, however, it's a pretty weak correlation. I think this graph really illustrates the idea quite well. that while pedigree is not a perfect predictor of genetic similarity, it does give you a pretty good ballpark idea of what to expect. You should just never put too much stake in that pedigree information. I just want to share one instance where the results are somewhat unexpected based on pedigree. So Georgia 10T is circled in red, is a cross between Georgia O1R and Georgia O2C indicated with red arrows. Grandparents were Georgia Brown and PI 203395 on the Georgia O1R side, and Sunbelt Runner and Georgia T2636M, which was a mutant selection from irradiated Georgia Runner on the Georgia O2C side. The distant relatives on this dendrogram are shown with the pink arrows. Given that 10T is made up of parents and grandparents from each of the three major runner clusters I mentioned earlier, it has a fairly diverse pedigree. It's still highly similar to Georgia O1R, which is just very unexpected, and it just goes to show that directional selection has a really big effect on overall inheritance of loci. So it's just important to remember that. To summarize this study, GBS was very useful for distinguishing between the elite set of cultivars we used, even some of those that were full sibs or reciprocal crosses. Within line variation was minimal, which was a good thing to see. And this analysis provides an in-depth understanding of genetic diversity and relatedness among the Georgia cultivars, above that which pedigree alone is capable of. And this study really makes available this genomic information for future crossing decisions and sets the stage for genomics-assisted improvement. So next steps include using this information to guide future crossing decisions and continuing to add to the data set with new breeding lines, external germplasm, and obsolete germplasm. And then hopefully someday making crosses to quantify genetic gain based on these distances. So I want to take this opportunity to thank our funding agencies, the Georgia Peanut Commission, Georgia Seed Development, and the University of Georgia Research Foundation for making this study possible. And I encourage you, if you have any questions or comments, to please shoot me an email at my email address shown there on the slide. Thank you very much.